much for the invitation and the opportunity to talk a little bit about Neurotez and what we're trying to do. The presentation will refer to our flagship drug candidate that is addressing Alzheimer's disease. I am Nick Tezapsidis, the president and CEO of Neurotez, which is a private biotech company. And I'm going to talk about Memtin, which is effectively leptin, as many people know it, our patented hormone replacement therapy for slowing cognitive decline in Alzheimer's disease patients and other dementias. The Alzheimer's disease is a huge problem in comparison. Coronavirus crisis that we're experiencing now is a midget. I say that because more than 5 million Americans are already with Alzheimer's disease, more than 30, 40 million worldwide. And it's the most common form of dementia. It gets progressively worse. There's no remission. The 6th leading cause of death in the US. And it's the only one out of the top 10 in America that for which we do not have a solution for prevention, cure, or even slowing down. It's that bad. The last drug approved for Alzheimer's was 15 years ago, and it only provides limited symptomatic relief. The figures are really, as outlined in this slide, provided by the Alzheimer's Association. Just a few to mention. It is expected by the year 2050 there'll be 14 million in the US. It is grossly underestimated how many people do have Alzheimer's due to the fact that a lot of seniors do not actually go and get a cognitive assessment, even though that most of them will agree that it is very important to have one. And it is costing a lot of money. In 2019, 290 billion to take care of these patients, and it is expected to rise to 1.1 trillion by the year 2050. And if you look at it at a global scale, the World Health Organization provided this slide. Alzheimer's disease is a form of dementia. It is the most common form. 60 to 80% of all dementia cases are the Alzheimer's type. However, you can get dementia from other causes like stroke and head injury, etc. But overall, dementia will cost by the year 2030 $2 trillion to take care of those patients. It's a huge unmet medical need. It's something that, and not only Alzheimer's, the other causes of dementia are also suffering from the same unmet medical need. So we've had tremendous failure rate in the Alzheimer's disease drug development. One of the reasons I believe is that Alzheimer's disease may not necessarily be a single disease, may be a multifaceted disorder, and we will be well served if we find ways by which we can identify subgroups that are a little bit more homogeneous in nature, both in terms of the phenotype of those patients and the genotype and the combination of the two. In order to identify those subgroups, we're going to need very sensitive and accurate diagnostics that will classify preclinical MCIs or mild cognitively impaired individuals and dementia due to Alzheimer's. And surprise, each one of these phases may need different approaches in order to provide clinical meaningful efficacy. And we are very adhered with those principles, so much so we're kind of in line with P4 medicine introduced by this guy here, Hood. I have his book there, which refers to predictive, preventative, personalized, participatory, and we add precision. You need all those P's in order to be able to address in a meaningful way Alzheimer's disease. And I'll describe to you what we're trying to do. So we're developing leptin, and this is based on more than 10 years of in vitro and in vivo preclinical studies done by Neurotez, confirmed by other independent groups around the globe. In addition to the preclinical work, there's some human studies, both retrospective and prospective in nature, and a small number anecdotal interventional human studies collectively supporting a role for leptin in neuroprotection, cognitive enhancement, its ability to decrease levels of phosphatidylcholine and A-beta, and these were the targets of other programs that failed in the past. And it is associated epidemiologically with lower risk for dementia in elderly. So we're offering or proposing a replacement therapy, a leptin replacement therapy, as relatively de-risked, because we have a very good picture of leptin safety profile, multifunctional because it has some metabolic activity, but perhaps it's not the most prominent biological activity of the protein, and a therapeutic approach that will contribute in reducing the rate of decline in Alzheimer's and optimally targeting early stage. I think everyone in the field agrees the earlier you start, the more likely you'll be successful, with characterized by hyperleptinemia. And I'm going to go back to the particular prodromal AD group, which is also by nature very heterogeneous, and the way by which we're going to address this heterogeneity. Now, leptin, most people know that it was a drug that originally was tried against obesity, primarily because the identification of the defective genes in the obob mouse, which this particular mouse has 2 leptin copies that are deficient, leading to this hyperobesity profile. In addition, however, to this hyperobesity phenotype, these mice are also characterized by a reduced brain volume and behavioral abnormalities, all of which can be corrected by supplementing leptin. However, the brain aspect, both function and structure, was not addressed ever by any particular pharmaceutical group, to our surprise. And this is something that we, one of the things that ignited us to try and use leptin in biological systems addressing Alzheimer's disease. So we are repurposing leptin. Leptin, in fact, has been approved, and it is in the market addressing generalized lipodystrophy, a super orphan, a rare orphan indication. So we're, repositioning or repurposing leptin. It's not strategically a bad path, even though it has resistance by big pharma due to the IP, etc. That may be weaker compared to a new molecular entity. However, big drug programs, some of them were much bigger opportunities in the new indication after repurposing. I'd like to mention aspirin, minoxidil, thalidomide, the compound that made Celgene, that started off, causing all those baby defects, is now a prominent compound addressing leprosy and multiple myeloma. And we would like to add leptin into that list of drugs that the new indication is going to be a much bigger opportunity. The advantages of repurposing a drug already in the market, you can take data that would speed up the process of both the preclinical stage and even the safety studies. We have good data about what we can use as a route of administration, frequency of administration, the magnitude of the dose, PK studies. The only thing that's missing from the safety profile about the leptin is the geriatric patients. It hasn't extensively been studied in individuals over 65 years. So we may have to do some safety studies there. The next few slides, I'm going to give you some of the data, the compelling data that are supporting leptin as a potentially very promising drug candidate for Alzheimer's. Starting off on the left by observational studies, this is a study led by Boston University and Shishadri. They followed 785 individuals from their elderly age that were at the beginning, at baseline, with no cognitive defects. And they demonstrated that those that had the lowest quartile leptin levels at baseline were 4 times more likely to get Alzheimer's 10 to 12 years down the road. That was the length of the study. On the right, there's a graph that indicates that perhaps hypoleptinemia may be somewhat specific for Alzheimer's, as in this small number of patients comparing Alzheimer's to vascular dementia patients with similar BMIs, leptin is very sensitive to BMI values. There was, again, a significant difference between Alzheimer's and vascular dementia, significantly smaller in Alzheimer's cases. From preclinical studies, leptin resulted in neuronal cells, and we studied 3 different cell types, in a dose and a time-dependent fashion, the reduction of A beta. And here, I'd like to make the statement, we do not necessarily agree with the majority of the investigators out there that A beta is the culprit of the disease. However, we totally agree that it may be a very significant biomarker that may help us monitor the efficacy of particularly disease modification entities. Along with A beta, we also studied the impact that leptin treatment had on phospho-tau levels within neuronal cells. Again, leptin reduced in a dose and time-dependent fashion the amount of phospho-tau within neuronal cells. And we have demonstrated this in different phospho-tau epitopes relevant to those that are found in Alzheimer's disease cases. Now, we did animal studies in 2 different models, both expressing human APP. And the first one is the, excuse me, the TG2576 that has one familial Alzheimer's disease mutation, and the other one is the CRND8 mouse, which the results of which I'm showing here. We confirmed, first of all, the data that we got from the cell cultures, where I was able to see that leptin reduced the amount of beta and phospho-tau. But going to the next step, we also demonstrated that leptin improved memory in this particular mouse. On the left, we see the novel object recognition test. Leptin, after 4 and 8 weeks, resulted in a phenotype or a performance of the transgenic mouse equivalent to that of the wild type animal, and vastly improving over the transgenic animal receiving saline. On the right, we have fear conditioning test, but in 8 weeks, both the contextual and the cued, where leptin again improved performance in these animals. In addition to these 2 tests, we did a number of other industry standard memory and cognition tests, and that leptin in all of them provided a signature indicative of pro-cognitive properties. And I mentioned the anecdotal studies in humans, and there were at least 4 studies done with a very small number of patients in each. All were led by Giulio Licinio, who is now the dean in SUNY, New York, and is member of our advisory board. Leptin replacement increased gray matter concentration in hypoleptinemic adults. These are not adults that had Alzheimer's disease, I have to mention. They were just hypoleptinemic and had a gross metabolic profile. The this phenomenon was plastic in nature. When you withdrew leptin treatment, gray matter concentration reverted back to the original, and vice versa. And most importantly, they treated a young hypoleptinemic that had cognitive development issues at 5 years of age. After 2 years of leptin treatment, he was able to catch up developmentally and is now a normal adult who very often calls Dr. Latino is very grateful for what they've achieved for him. Linking leptin levels and Alzheimer's disease, we took advantage of the measurements of leptin in serum and CSF that were done in a subgroup of patients that had enrolled in the ADNI study. Alzheimer's disease neuroimaging initiative is a consortium of academic and pharmaceutical companies, consortium that addresses biomarkers in volunteers with or without Alzheimer's disease. And a subgroup had leptin measurements done. We were, excuse me, we confirmed the link of low leptin and Alzheimer's disease, both men and women. I specifically address men and women because women generally had 2 times the levels of leptin compared to men. But in both genders, approximately 70% of mild cognitively impaired men and women had leptin levels that was lower than the median leptin values found in normal elderly. So a significant portion, and this is cross-sectional in nature, okay? A significant portion of MCIs and also AD, which I'm not showing here, have low leptin levels. And a significant portion is concentrated in what is the lowest quartile in normal. Here in men, about 45% belong at a level of leptin that is in the first quartile, and about 30-35 for women. So what's the mechanism of action, one would ask, and how come leptin, which is a metabolically active compound, is going to be of benefit for Alzheimer's cases? What we do know is that there is a high density of leptin receptors in the hippocampus. We also know that leptin readily transports from the periphery into the brain. There's a saturable transporter that will allow this to happen. In fact, there's a complete correlation between the levels of leptin in the periphery and the CSF. So whatever reading you get from plasma, you'd be able to translate it in leptin levels in the CSF. Now, activation of the leptin receptor would activate, according to our data and looking at other reports, AMP-dependent kinase, GSK3 beta, and SIRT, all of which are somewhat directly or indirectly linked to modulating A beta and phospho-tau. And this is why we observe change in the levels of A beta and phospho-tau due to leptin treatment. It's done through molecular centers like GSK3 beta and AMPK. In addition, another group has indicated that leptin has an impact in modulating NMDA and AMPA receptors. And as we all know, these systems are usually targeted by the most recent compound approved for Alzheimer's, Namenta. And so collectively, these functions should provide the basis for leptin's ability to improve plasticity and synaptic function, decreasing the rate of decline in dementia. A lot of people have referred to Alzheimer's disease as type 3 diabetes or the diabetes of the brain. And this graph indicates that there is substantial overlap in some of the pathways between activation of the insulin receptor and leptin receptor. And leptin is known to be somewhat a sensitizer of insulin action. And I'd like to refer to the programs led by GSK and Takeda that involved insulin sensitizers, the pioglitazone and rosiglitazone in Alzheimer's trials. So that we are not the only crazy group out there believing that metabolic pathways are important in Alzheimer's disease. There's a big, substantial portion of research that follow and believe that metabolic pathways are very important. And as I mentioned to you, leptin activation of the leptin receptor through AMP-dependent kinase, which, by the way, is regarded by many as an energy sensor, not only of peripheral cells, but also neurons. And you can understand how activation or modulation of AMP-dependent kinase as a target of leptin can be a meaningful target for other therapeutic interventions leading to its activation or modulation. So this is a novel and differentiating approach. Type 3 diabetes for Alzheimer's is not an uncommon characterization. It is a natural protein that has procognitive properties with known to be, in a good portion of Alzheimer's case, at low levels. We know that ameliorates A beta and tau pathologies. So the rationale is it is modulating an upstream common molecular target. So the clinical strategy that would involve an enrichment of patients targeting those that are low leptin at the early stage and genotypically predisposed, and for that aspect, we are collaborating with Cytox, a UK-based company that has tens of thousands of SNPs that they use to detect the presence of absence in combination with an algorithm that will determine risk for Alzheimer's disease. We're very excited about this approach in identifying the right target. And I'd like to mention a little bit about the previous failures. I think the antibodies failed miserably, and I still don't understand why this is explored as a drug candidate, even if there is, and the possibility here is remote, a chance of getting anything meaningful by removing A beta with antibodies. It's not going to be a particular patient friendly. It's not going to be cheap, and it's simply something that will not. Also, given by if it was efficacious, I can understand. But even with the latest data from Biogen, it's not particularly impressive. I don't know how this will do with regards to insurance companies and the payers. So it would only be afforded, and for a glimpse of hope, by very wealthy individuals. So another reason for failure was the heterogeneity in patient groups recruited in Alzheimer's disease trials. And most importantly, the A beta and phospho-tau are more than likely the wrong targets, period. No BACE inhibitors, no gamma secretase inhibitors, no antibodies against them are going to be ever successful. So another unique aspect about our approach is that we can potentially qualify for an accelerated approval using leptin as a surrogate biomarker endpoint. But for that, we will have to demonstrate on a phase 2 that we can achieve not only a shift in relevant biomarkers, namely A beta and phospho-tau, but also provide a cognitive benefit. And in order to do that in relatively short trials, perhaps the first type of patients that we're going to be addressing will be slightly more advanced than the ideal target that we have in mind in the future. Sorry. So we're relatively, even though we regard ourselves as phase 2 ready because it is a repurposed drug, therefore we have all the safety data available to us. The company is in the preclinical stage. We will still have to manufacture the protein. I have to do the IND enabling studies before we do the phase 1, relatively perhaps short, and then straight into a phase 2, looking into biomarkers. I mentioned, I'll show you the next slide. I mentioned that perhaps our first phase 2 trial would involve later stage individuals that would enable us shorter preclinicals, shorter phase 2 trial data, and perhaps strategize in order to make this trial sufficiently strong to lead towards an accelerated approval. It can even be under the accelerated approval program, a trial that we can use to get conditional approval to be followed by phase 4 post-marketing studies. I'd like to mention some of the team members. These are top-notch leaders in academia and biopharmaceutical industry. MacArthur has been more than 30 years with Pfizer, dealing with biologics. Dr. Ashford, he's the has been the CMO or acting CMO almost since the inception of the company. Has been working in the field even before the first cholinesterase inhibitor was approved by the FDA. George Perry is another prolific key opinion leader, our chief scientific officer, who has more than 1,000 publications in the field. He's an editor of a journal, and is a well-respected and has a lot of citations, thousands. Jukka Karjalainen is an experienced drug developing individual. He has been working for small and big companies, dealing with drug development efforts from phase 1 to phase 4. James Harris has also experience with biologics and biosimilars, originally from Amgen, and then developed his own company. Michael Hoy, a regular affairs guy, more than 15 years in pharmaceutical industry, works now with big pharma. He's only a consultant because he's restricted in being more than that. Jane Johnston, VP of Operations, has years of research in the field, in the neurosciences, and is currently in academia helping us with our project.